This is one of the internet's favorite cell videos. It's a neutrophil chasing a bacteria around until it finally eats it. But did you know that cells can eat other cells too? The big eaters of our immune system is the macrophage. Macro meaning big, phage shortened from the word phagocytosis, which means the process of eating. Macrophages are the cousins of neutrophils. They're a bit slower at chasing down bacteria, but boy are these guys the super eaters of our own cells. Our body can be thought of as a giant ecosystem made up of tens of trillions of cells. But every single day, about several hundred billion to 1 trillion cells in our body are dying. While that's totally normal as old cells die and get replenished by new cells, it does mean that there's a ton of cell corpses that need to be disposed of. When a cell dies, it pumps out several types of signals, the find me signals and the eat me signals. Both are very potent at attracting macrophages. One of the best studied eat me signals that physically links a macrophage to a dying cell is a lipid on the dying cell surface called phosphatidylserine or PS. A cell that is happily alive has PS facing the inside of the cell, but a dying cell will have PS flipped to the outside, catching the macrophage's attention. This externalized PS in turn can bind to several receptors on the macrophage surface, where these receptors will work together to pull the dying cell corpse into the macrophage. This process of a phagocyte or eater cell enveloping and digesting the corpse of a dying cell is called efferocytosis. Since it's estimated that about 0.4% of our cells, or 1 in 250, are dying on a regular, healthy day, a macrophage in the body will stuff themselves with dying cell corpses so as to keep our tissues and organs clear of debris. But scientists also think that eat-me signals like phosphatidylserine, along with the efferocytosis process, are ways to dampen the immune system, to prevent chronic inflammation or autoimmunity. One way that this happens is through surface proteins like PD-L1. PD-L1 is the counterpart that fits with a protein called PD-1, which is displayed on the surface of killer cell types of our immune system. The physical linkage between PD-L1 on the macrophage surface and PD-1 on, for example, the T cell surface dampens the activity of the T cells, making them tolerant to their surroundings. The scientific term for this process is called immune tolerance. Within a tumor, cancer cells will opportunistically hijack this trait of macrophages to their advantage. When cancer cells die within a tumor, the same process occurs where their corpses are efferocytosed by macrophages, who in turn starts to display PD-L1 on their surface. Now, when patrolling T cells that are supposed to be attacking the cancer cells come by, their PD-1 encounters the PD-L1 on the macrophage surface, and these cells get tamed and are no longer doing their job to kill the cancer cells. This type of immune tolerance allows the cancer cells to grow unchecked by our immune system. So scientists have given these immune dampening signals like PD-L1 a new name, checkpoints. One question that sprang forth naturally is then, what if we blocked the physical interaction between PD-L1 on the macrophage surface and PD-1 on the killer cell surface? Would that unleash the T cells and other types of killer cells to do their jobs again? This concept became the basis behind what we now call immune checkpoint inhibitors, or ICI. Simply put, these are antibodies that bind to molecules like PD-L1 to try to block these molecules from interfering with T cell function. The first ICI therapy came into use in the year 2000 for metastatic melanoma and ovarian carcinoma. As of the making of this video in 2023, ICI is being used in clinical trials for over 50 different cancer types. For metastatic melanoma, which was the first FDA-approved cancer type for ICI usage dating back to 2011, the response rate to date has been promising, with 15 to 20% of patients achieving complete response and living cancer-free after the end of treatment. But that also means that a substantial proportion of patients treated with ICI do not respond, and many discontinue treatments due to adverse effects. In fact, the response rate to ICI is very different between cancer types and even among individual tumors within the cancer type. Figuring out biomarkers and metrics that could accurately predict the response to ICI so as to determine which therapy to use to maximize benefit while minimizing harmful side effects is critically important. and will help significantly in the development of personalized precision immune oncology treatments down the line.